In this video, we will discuss about tumor-derived lymph node role in tumor development. So we have discussed these things in our previous video also. However, we will continue our discussions and we will discuss some of the in vivo results. So before going into the in vivo results, let us go through the results that are being tested in different cell lines. So as we know that this capsaicin or RTX based induction that prevents the TAM, that is tumor associated macrophages interactions with this tumors. In that case, they are able to they see the effect of this TAM preventions, DM interactions that are being prevented with the tumors on the tumor volume. And in the previous videos, we have see the effect on the mechanical sensitivity. Over here, we are seeing the effect on tumor volume. So when this RTX based treatment is done, that prevents the TM interactions with the tumor, we are able to see a reduced tumor volume, indicating that TM interaction promotes the tumor volume. That is validated in the control. And these things they have validated in the 4 different cell lines, that is head and neck squamous cell carcinoma, melanoma cancer, fibrosarcoma, and also fibrosarcoma, but these are 2 different cell lines. And in this case, they are able to see that tumor volumes are suppressed in all the cases. So this is also the in vivo studies where they did the where they inject these cancer cells in the mice. So in all the 4 cases, they are able to see tumor suppressions when this capsaicin or RTX based treatment is done to prevent TEM interactions with the tumors. And these things they have further quantified in the panel B, where we can see that in the case of SAM surgery, there is a tumor suppressions when the RTX or capsaicin based treatment is done. And thereafter, they check it for the other system also, where what it is, they transduce this Scn10a CatCh-EYFP into the tissue, which is optogenetic based activations of this CatCh, and this optogenetic based activation of the CatCh will promote the tumor volume. And this is the things they, and this is the same thing that they have seen in the B16F10 transduced mice model. There is an increase in the tumor volume when there is optogenetics based activation of this CatCh occurs. However, one more thing that they have observed, when they remove the cervical lymph node using cervical lymphadenectomy, in that case they are not able to see these changes for both the cases. That is, RTX based treatment does not show any change in the tumor volume between the control and the RTX based treatment in the absence of lymph node. And similarly for the B16F10 also, they are not able to see a change in the tumor volume in the case where tumor derived lymph node are absent. And in the panel C, the treatment is done in which this optogenetic based activation of the CatCh enzyme is carried out. But here also did not see any change in the tumor volume, indicating that lymph node is necessary for this tumor derived lymph node is necessary for the tumor development or nociceptive related tumor development. Thereafter, they did some more mice studies, or they checked the other systems that they have validated in vitro studies, where what it is, they compare the RTX based tumor suppressions with this HM4 DGI based tumor suppressions in the presence of DCZ. So what they are able to see that RTX based tumor suppression is less pronounced compared to the HM4 DGI based tumor suppressions in the presence of DCZ. This is true for these B16 F10 cancer cells and SCC7 cancer cells, and or they can check it, they checked it in the 2 cancer cells, and in both the cancer cells they are able to find the same results. Afterwards, they checked for the other system, that is CatCh based optogenetic activations, whether these things can be the tumor progressions can be reversed if what it is, they did the same things, that is HM4 DZZ base transductions along with this DCZ injections or inoculations using drinking. In that case also they are able to see that when only the CatCh optogenetic based activations are there, or when only the CatCh is transduced, they are able to see enhanced tumor volume. But when they also transduce HM4D GA, the tumor suppression is rescued and they are able to see tumor suppressions close to the wild type conditions, and similar results they have seen for MC5-7G. So this in all together indicates that all the three systems works, and the presence of this tumor derived lymph node is necessary in order to act on this tumor development, either for the tumor progression or for the tumor suppression. That's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.